We've developed a world-first technology that allows us to isolate pure transcription factor, in this case CTCF, DNA from blood samples. That means we can isolate pure DNA that comes from a cancer, and that gives us a lot of potential applications using a completely new class of biomarkers. And of course it's a new method for liquid biopsy, which is revolutionary and doesn't follow the usual pathway, which is that you simply extract all the DNA and sequence it.
I'm really excited about this breakthrough that Volition has come up with, because we are really shifting the paradigm from searching for a needle in a haystack to literally removing the haystack. This is really important from a clinical perspective for patients, because imagine that you're panning for gold. Most of the liquid biopsy companies are scanning through the entire riverbed. What Volition has managed to do is almost have a magnet that pulls out the gold from the riverbed before you even start counting it, or in our case, sequencing it. No one ever really thought that this kind of purity could ever be achieved from immunoprecipitation, and Volition has managed to do it.
Capture-Seq aligns with Volition's core principles and core purpose, which is to bring tests to patients to improve the speed with which we make diagnoses and facilitate treatment. Diagnosing cancer early will improve outcomes, and also for healthcare systems too. It means less surgery, less therapy, less treatment. Capture-Seq genuinely is a methodological first, being able to isolate tumor-specific cell-free DNA, and we really hope that it will be transformational as it moves forward.
What Volition are doing is they use a chromatographic process whereby they use antibodies against a well-known transcription factor called ctCF to pull out sequences of genomic DNA which is floating in the circulation, which is attached to these transcription factors. And that allows an enormous enrichment of these particular sites in the genomic DNA. And it turns out that these sequences occupied by the ctCF transcription factors are very prominent in many types of cancers. Surprisingly non-intuitive is the fact that these sequences, of course, are in every cell, not just cancer cells, such that when the DNA of any sort is released into the circulation, the factors only seem to stay bound to the particular sequences of DNA in the circulation if they have originated in a cancer cell. So this enrichment removes the signal to noise, which besets all types of genomic analysis of cell-free DNA in the blood, such that it gives a very strong signal even though there's very little of this circulating DNA. And so that's what's described in the manuscript. And what we now need to do is apply the technique to much greater numbers of blood samples and just get reassurance that not only does this detect late-stage cancers, but very early-stage cancers as well, which is a real unmet clinical need in this area.
I'm convinced that it will shake the current DNA analyses, so it really goes beyond what we are currently using today. The versatility of the platform will also allow to apply to different applications. So not only cancer diagnostics, but we can probably be able to discriminate between cancer and inflammation and all types of different pathologies. We've been testing cancer samples from stage 1 to stage 4, and when you look at the ability of the platform to discriminate between those conditions, it's just phenomenal. The team, we're currently looking at optimizing technology already to make it quicker, more affordable, and for it to be used on a day-to-day basis by a multitude of different groups.
So I'm very excited to start a round of discussion with potential commercial partner about this new technology, which has a very large range of potential application, from tumor-naive MRD to early cancer detection. And what it's worth mentioning, it's both on the human side, obviously an enormous market, but also on the animal side. It's a new class of biomarker that fits very nicely into the portfolio of both those human large, liquid biopsy companies, as well as the existing portfolio partners that we have on the animal side and new ones that we're also in touch with. So we have started commercial discussion. We are getting a lot of interest, and we expect this interest will materialize into commercial agreements later this year.
What makes this so exciting is this, we believe, is the first true pure liquid biopsy. We're getting 99.99% of the sample is pure tumor-derived DNA. This is truly remarkable and a fantastic breakthrough, but we now have a complete package of technologies from our nucleosome packages and now with capture in transcription factors. And I think it's a package which will help revolutionize diagnostics in a very wide range of areas.